is a relatively short section about the workflow overview. Right, so I'll just go through either one. So since the base-based configuration is more complicated versus the standalone configuration when set up in your run, so I'm going to go through this one. So the first step is to prepare libraries, and you have to enter the run parameters in the base space before you actually start the run. So if you want to connect the run to the base space, what you need to do is to input all those sample and index information in the base space first, and then you set the run as run ready for sequence. And after you go for the NextSeq and then you log in your base space account, it will automatically pop up all those runs that are ready for sequence from your base space account, and then you can just go through and set up the run. So before that, you need to set up the run parameters in base space. So for those who are selected to be involved in the hands-on section, we will work together this afternoon on how to set up your runs on base space. But I will also introduce briefly in this presentation. So after setting up the run, and actually I suggest you to prepare the runs before you prepare your libraries. That's because it's always better to prepare beforehand so that you have a very good index combination before libraries actually constructed. Because you have to think about the index combinations and not overlapping index for different samples. And for enrichment, sometimes you pull a lot of samples for one single enrichment, and there is no way that you can separate them anymore after the next one. So prepare that and plan your experiments before actually you set up the runs. And after prepare libraries, so we'll prepare all those sample information in the preptet. So preptet is one of the functions in the base space, and is specifically designed for NextSeq system, where you can enter your all sample informations, including the index. And if you want to connect the base space, your data to base space for the multiplexing, then it's essential to prepare the preptet in the base space account. But say if you want to do it as a standalone, then you don't need to care about all this, because you just need a sample sheet after the run to complete, and then you prepare the multiplexing. Okay? And so this is the screen cap of the preptet. So after you log into your base space account, so how many of you have already registered base space account? 1, 2, 3, 4. Okay, so you just go to My Illumina, and then you register to My Illumina account. Then you will have access to base space and other algorithms online, such as the Design Studio for designing your own custom panels. And after you log in, you just click the preptet, and there are several steps. The first step is to click biological samples, and then you enter your samples, and then you prepare your libraries. Then you can choose the library prep kits. So almost all Illumina supported library prep kits are pre-entered in the base space preptet. But if you are using custom libraries, you can create your own libraries on the preptet. I will demonstrate how to do it in the afternoon session. For Gordon's run, so today we are going to start with your own libraries. I have checked the index sequence, which is essentially the same as our TrueCLT library kit. So we can use this as a template. So after choosing the kit, it will pop up all those index names, and you can just drag your samples. Say, for example, sample 1 is index A002. Then you can just drag here, and then you will essentially pop up the actual sequence of that index, and then you can have a double check here. Okay. So if custom library package, you just choose custom library package, and then you can download a template, and is to open the template in Excel, and then you just change the pre-enter index name and the sequence, and then upload again, and then give it a name. Then it will display in the list of your basic account. So next time you don't need to do this again. You just choose the one that you have named for your customer account. And remember, when labeling or samples, we only accept letters, numbers, hyphen, and underscore. Do not put in other characters because it's not going to recognize, and you pop up an error during the analysis. So after you choose the key and identify the index sequence, and then you click next, and then it will ask you. So this is your library custom library prep key. You can also give it a pre-designed sequencing name, whether it's single repair n, is a single index or dual index, and how many cycles do you want to do in 2 different weeks. And this is where you can download the template, and after modify the template, just choose your amended template, and then you can just upload. So that's quite easy. Okay. And after that, you can go for the next one. So the pools. So here is where you want to put libraries. Sometimes you will prepare, say, a whole plate of libraries, say 96 samples, but you may not sequence the 96 samples in one single run, right? So you can just choose from here, so which particular sample you want to put in this run. Or even though sometimes you want to repeat the sequence for certain samples, you can come back again and then pull the samples again. So you say, I want to pull, for example, here I have prepared 4 samples. If I only want to sequence number 1 and number 2, I just drag number 1 and number 2 to here, and I give it a name, and then you click plan 1. So after choose the plan 1, it will ask you to give a reagent barcode. So the reagent barcode is the barcode in the reagent cartridge. But I suggest you to leave it blank because sometimes when during the process that you're setting up the prep tag, you're still not sure which particular kit you are going to take out, right? So just leave it blank, and then you can modify the cartridge during the run setup. And because if the reagent barcode that you enter is different from the actual cartridge that you're going to use, the instrument cannot identify this run from your base account. Okay? You cannot. So you just leave it blank. And if you use custom primers, you have to tell. So give it a check if you use custom primer with 1 week to an index. And enter whether it's single repair and how many cycles. So here, the target is 2 times 150 for the insert size, but I want you to put 1 more cycle for some calculations that I'm going to mention tomorrow. And then you click sequence. So after you click sequence, then this run is ready to sequence. If you keep it in this page, then the instrument cannot recognize it because it's just in a planned status instead of a status that are ready to sequence. So say if you leave it blank but you still cannot recognize from the instrument, then probably you want to come back to see whether the run is still in a planned status or is already in the sequence status. Okay, is
so sorry, so for this step I can do it in my computer, right?
Yes.
And how about can I do it on site on the sequencer?
On the sequencer, then if it's connected to the internet, then you have to log in base account to it.
Just like a normal PC.
Yes. So if you don't want to connect to base, then that's even simpler. During the setup, it will just ask you, are you going to do single repair and what is your sequencing name and whether custom index primer are required. Okay. So library preparation thing we have just mentioned that, and Aaron is going to give you a little bit more detail on that. So I'm not going to go over details, but you can check for this link. We have the library prep kit selector, helps you and guides you through to choose the right kits to use. So you can also import your library. Say, for example, I have 100 samples, 100 libraries. Do I need to enter each samples in the biological sample step, each one by one? That's quite tedious. So if you have lots of samples, you can actually import from a template. You can download the template and then you enter all those sequence and sample names, and then you can just import all those in the template. Okay. So after preparing the peptide, we come back and then we will denature the library with sodium hydroxide, and then we will, after the denaturation, we will neutralize it with the twist HCl buffer. So this is trying to minimize the effect of sodium hydroxide. So the target is that we need the final library, ready to load library, with a sodium hydroxide concentration less than 1 millimolar. Because if the sodium hydroxide concentration is too high, it will affect your customary efficiency because it's denaturing agent. Okay, so we'll denature, neutralize, and dilute with the HT1 buffer. The buffer is available from the accessory box provided from the kits. Okay? And then we'll prepare the reagent cartridge. So for the reagent cartridge, either the night before, you can thaw it at 4 degrees overnight, or at room temperature water bath. It takes around 1 hour. And prepare your flow cell. So bring the flow cell to room temperature for at least 30 minutes. So, yeah, we want to avoid any condensations, which will affect the imaging. And then we load the libraries, and then we can start the work. Okay, so the target cluster density is 120 to 30 K per mm square, and we need the 200 millimole twist buffer for neutralization. And we also need sodium hydroxide for denaturing. And the recommended loading is 1.8 picomolar in 1.3 ml of libraries. So it's very little amount of libraries. But this is based on the 5X libraries. So 5X library is a prepared library available from us, and the medium size of that is around 500 to 525 base pair of the 5X library. So if your library is a bit shorter or longer, then it's a possibility that you probably need to adjust that, because we know shorter libraries custom more efficiently than long libraries. Oops. Okay, so and then before the reagent cartridge, so make sure it's fully full, and then we want to mix it, invert up and down 5 times, and then we want to tap on the surface to remove bubbles, and then dry the base of the cartridge just to ensure not to bring a lot of water into the instrument. This is the water line here. So when you try to thaw in a room temperature water bath, do not exceed this water line. Okay. And if you're interested on what reagents are pre-coded in the cartridge, you can refer to this table. I'm not going to go through details. This is what we loaded libraries, number 10, and custom libraries are 7, 8, and 9. Custom sequencing plans. So number 7 is for custom read-run primer, and custom read-to primer is here, number 8, and number 9 is custom index primer. So if you have both custom index 1 and custom index 2 primers, since there is only one well, then you're going to mix your 2 primers together. And the flow cell, again, take a room temperature 30 minutes before use. Remove the package. It's an aluminum sealed package, and you just need to clean it briefly with ethanol wrap, and then with Kimlite, then that's all. So if you open it and with no other reasons that you want to postpone your run, say, oh, I have some urgent matters I cannot settle in now, then you can put it back into 4 degrees up to 12 hours. This is still stable. And for the reagent cartridge, you can also put it in 4 degrees, but maximum is 16 hours. Okay, so if you already thaw it the night before, then you better stop the run. But if you just throw it today, then you can still keep it for a few hours. And before loading the flow cell, I suggest you to check the integrity of the flow cell. So what I want you to see is to ensure that the foam metal plane is flat and unclip the flow cell tightly. Okay. And we want to check, so here maybe you can't see properly here, but here you can see. So this is a gasket. You can see there are 2 holes. 1, 2, 3, 4, 5, 6, 7, 8. So these 8 holes are essentially where the fluid is flowed into the flow cell. So ensure that these gaskets are free of obstructions if they're not blocked. And you can check also this white plastic is in place. So if say there is one that have a different level, then you probably want to manually clip the flow cell to ensure that they're in place. Okay, this is the gasket. And then you just load the libraries. So for earlier NCS version, the loading concentration volume is a bit different. But for you, since the version that currently installed in your NextSeq is version 2.0, so you're going to use one on CME. So for earlier version is for earlier version 1 reagents, but that's discontinued already. So what you're going to get is version 2 reagents, and you need version 1.4 or above softwares. Okay, and then just very easy, click sequence in the NCS, and then after loading all the reagents, it will guide you through. So this is the standalone mode of the instrument. So you can choose pair N, and then you enter number of reads, the read length, and whether you want this custom timer. If you already set all those in the base pattern, then it will display automatically according to your setup. Say for example, from here you just see, oh, I typed something wrong in the base. You can actually click here and edit it from the setup page. And after you click start the run, click next, the instrument will check itself for a pre-run system check before it starts the run. You can actually skip the pre-run check by setting some parameters in the manage instrument, but I do not recommend you to skip it because the pre-run check only takes about 2 minutes. Okay? So it's always best practices to let it do a pre-run check. And then after that, you just click start, and then it will do the analysis. Okay. This is just a software compatibility issues, but I don't think this is an issue anymore because it's during a transition state that we have customers running both version 1 and version 2 reagents, and they have some software compatibility issues. But since now version 1 is discontinued, then this problem is gone. Okay, so and then finally procedures. Post-run wash, and the instrument will do a post-run wash automatically, and what you do just remove the reservoir, and then manual wash is required every 2 weeks only. Okay, and check the disk space, and you can just leave the instrument on at all times. So after each one, you can just leave it on. But if you want to shut down, of course, you can also do a shutdown. But before the shutdown, do a manual wash. Post-run wash, you will do the post-run wash automatically, and it takes around 90 minutes. Okay. And after completion of the run, for waste disposal, the reagent cartridge, the only well that you are concerned may be is number 6. Position number 6 is a LDR buffer which contains formula, and you need to dispose properly. And the design is that after the run, the instrument will suck up all the remaining reagents to the waste buffer. So the remaining reagents left over should be very little, but if that's still a concern, you can actually detach this well away. Then you trace this mechanically. But having said that, that means the waste buffer is contaminated with formula and needs to be disposed as a chemical waste. Okay? And also, manual wash is required every 14 days. Okay. To perform a manual wash, you just click perform wash, and then it will ask you to load and use the flow cell to wash. So we need a use the flow cell to perform the wash, or with the instrument we have a wash flow cell come along with the instrument. You can use the wash flow cell as well. Okay. And just follow what the instrument tell you, and then we'll have a wash this afternoon, so you have got a chance to do that. And it takes around 20 minutes to complete. So the wash buffer needed is 0.05% of 2020. So this is quick wash. That means it's the manual wash every 2 weeks. And manual post-run washes in general is not required because the instrument will perform a manual automatic post-run wash. But say in some cases, like the run is aborted in the middle of the run and auto post-run is not washed, then you have to come here and then do a manual post-run wash. For a manual post-run wash, you need the 0.05% of 2020, but you also need 0.12% of sodium hypochlorite in position 29 to allow you to wash the 1089. Okay, so this is generally not necessary unless the run is interrupted for some reason in the run, or if when you have very high usage of the instrument, say you're running it basically every day, then I suggest you to keep additional manual post-run wash between runs, so that you can keep the lines of the instrument as clean as possible. And what is supplied with the instrument is a reagent, is a wash tray. So just like the reagent cartridge and wash buffer, and for a quick wash, you just keep this reagent cartridge empty. You don't need to put any wash buffer inside. You just put the wash buffer to this wash tray here, and the instrument will pump the reagents to the tray automatically. But for manual post-run wash, besides adding the wash buffer, the clean buffer here, you also need to add sodium hypochlorite to position 28. Yep. So that's the only two work that you need to do. And then after the wash, you just leave the flow cell, wash tray, and span trays on the instrument until you start your next one. So just leave it, that's fine. And you don't need to change the nickel first between the wash because the volume is sufficient enough. Okay. And finally, about the disk space of the instrument, it only contains enough disk space to hold data for one complete run. So for the highest output and 30 cycles of run. So as I just mentioned, suggest you to use the network drive for data storage. Okay. Drive. And for shutdown, if you want to do the shutdown, just do a manual wash, and then you tackle the main switch at the back of the instrument. And if you want to switch it on again, say for example the window hangs, then you probably want to force shut down it, and then you turn on the main switch of the the hardware, and then you need to wait at least 1 minute before you can switch on. So give it some time to cool down before activating it. Okay. And the best practices is that the RFID will take several seconds to register. So that slow mode, it just like it's handling right now, but it just takes some time to do the registration. So just be patient and wait for it to complete. And empty the span tray cartridge before each one. So the waste tray, you need to empty it. I guess for the high output 30 cycles run, the span tray is not possible to retain more than 1 ounce of reagents. So please empty that before everyone, and the software will guide you through to empty reagents as well. Okay. And ensure the bleach prepare is freshly prepared, and keep the wash tray clean after use. Invert them to let them dry to avoid the growth of most, and leave the flow cells and the instrument until the next run. Okay, and save data in the network drive, and you can keep it on at all times. Okay. And for analysis for different applications, we have different apps available in the base for analysis. I'll go through in details in tomorrow's base session. So this is just a highlight summary. Okay, and library prep, I guess Aaron is going to touch more. Okay, and finally for the NextSeq, you can share the run with your collaborators, and you can also share the run. Okay, so just give the email address and then click invite, then they can accept the run. Okay. So questions so far? Okay, if no, then I'll hand over to Aaron. USB.
Morning and Happy New Year. My name is Aaron. I come from the Illumina marketing department, who is in charge of the library prep product line in Greater China. So today, please allow me to use 1 hour, following 1 hour to finish this training session to introduce our products. And after this training session, you will describe the different library prep workflows from DNA, RNA targets, and some epigenetics library. And you will get to know the library prep best practice. So, a quick review as introduced from previous slides of Emily. Total workflow of next generation sequencing includes the library preparation, class generation, sequencing, and data analysis. So step 2 and step 3 can be finished on this machine. So it's kind of automatic step. And in this step, we provide a family of different softwares on the base environment. So they're very powerful for your data analysis. Here, I would like to emphasize the importance of this step, the library preparation. It's not just because my job is to promote this product, but because this step is much more complicated and to some extent more crucial than the other part, because you need, most of the time, you need to manually finish the library prep. So different to these steps, you can rely on our machines. This step, you should rely on yourself for the manual processing. For the library preparation overview, firstly, we need to get some extracted and purified nucleic acid samples, DNA, RNAs, and then we need to modify the nucleic acid samples to the proper insert size. And then we need to add the adapters with size 4 the adapters to our DNA or RNA insert. These adapters should have the size for the flow cell binding, the sequencing primer binding, and sample pooling indices. So these slides you have seen it before. A quick review is we should add 2 adapters at the 2 ends of our DNA insert. These adapters should have the sequencing primer, binding site, index, and the binding site on the flow cell. So no matter what is your input sample, the final constructed library should look like this. Looks like this. DNA insert together with the adapters. So here we can see there are quite a large number of options from Illumina. So we provide the library prep kits from the RNA, from the mRNA to the RNA, small RNA. to some epigenetic study and some different DNA library kits for large genome, small genome, short reads, and even some long reads. And the other fixed panel or customized panels for your target sequence. So we provide the library preparation kits across nearly all of the applications. As Emily mentioned before, we have some very helpful online tools. We call it Sample Prep Kit Selector. Just open Google and key in the kit selector Illumina, you will find that website. So that tool will guide you step by step. Firstly, you can select your application, DNA or RNA, and then you can tell the information to this tool about the genome size. For example, the species of your samples, something like that. And finally, the tool will guide you step by step, and finally recommend all of the appropriate library preparation kits. And then you can compare the hyperlink compare kits, and then it will show you a detailed table to compare the features of different preparation kits. And finally, you can select your preferred library kits, and it will guide you to a support page. From that page, you will find all of the information from the features to the quick guide to some detailed menus and even some comparison with comparison data or some demo data. So you will know all of the information. So it's a very helpful tool. You can try to use it. To simplify this session, I would like to categorize sort of our products into 4 groups: DNA library, RNA library, target library, and some other library preparation options. Here in these slides, I would like to highlight more on the RNA library preparation and the exome library preparation in this target library group. And firstly, maybe we can see the DNA library preparation options from Illumina. So here, majorly we provide 6 types of DNA library preparation products. The top 2 are based on we call it TrueSeq series. Based on the classical way to get the DNA fragment is use some ultrasonicator to generate some mechanical shearing of the genomic DNA to get some DNA fragment. The first way. The second way is utilize some special enzymes. We call it transposon. So this enzyme will help us finish the fragmentation of your genomic DNA. And the third options, we use these 2 kits to provide some long sequence reads. So for example, if your application is some de novo sequencing or some haplotype phasing study, this long sequence read will be very helpful for you to accurately construct the genomic DNA. So we have 2 different choices based on different technology. So I will go through that later. So firstly, let's see the TrueSeq. So TrueSeq is the most widely used application to prepare the DNA samples. We have 2 types of TrueSeq library preparation kit. One is we call TrueSeq Nano. The other one is TrueSeq PCR. Actually, these 2 kits, they are very familiar. So let's see the TrueSeq Nano. TrueSeq Nano, its feature, I'd like to emphasize this part. So it only requires 100 to 200 nanogram DNA per sample. So why we call it nano DNA. So this kit, we use the bead-based size exclusion, no use the gel any longer. So it only requires small inputs. So after the ligation of the 2 adapters, the library already been constructed and ready for the sequence. So the TrueSeq Nano and the TrueSeq PCR-free, they has been optimized for the high quality genomic DNA. And if you're very cared about the sequencing coverage uniformity or something like that, TrueSeq series will be your best choice. So the TrueSeq kit they are compatible with all the Illumina sequencing platform from HiSeq, NextSeq, or MySeq. And yeah, the second one is TrueSeq PCR-free. So PCR-free, they prevent the PCR step. In this way, we can prevent the bias from PCR process. So without PCR, the workflow is faster, half day, and it's also based on the base size inclusion, the gel-free one. And it will give you a very good coverage of at different regions. We will get smallest gaps. But without PCR, we need a bit larger amount of the input samples from 0.1 microgram in a nano kit to 1 microgram in a PCR-free kit. So similarly to the nano kits, we can also index up to 96 samples by pulling more as 96 samples in one library. Yeah, so it will also been optimized for the high quality genomic DNA and compatible with all of our platforms. So let's see the workflow of the DNA library preparation. Firstly, we get some extracted purified DNA, and then we use mechanical shearing. It requires some additional machine to make the fragment of the DNA. And then we need to use some reagents to repel the ends of each fragment to build a blunt end. And then add the phosphate at the 5' end. And then we use some bead-based size exclusion to get the size we need. For example, we need 300 base pairs, so it will in the peak across with small narrow region. And then we need to do some annihilation at the 3' end and finish several rounds of the bead-based size selection again. And finally, we will use some ligation enzyme to ligate all adapters at both ends of the DNA insert here. And for the nano kit, here we need to go through 8 cycles of the PCR to amplify our DNA fragment. Now after the normalization and the quantification, we can get the library ready for sequencing. So it's a brief workflow of the nano kit. As comparison, for the PCR-free kit, all of the previous steps are the same. Only difference here, after the ligation, we already get the library ready for sequencing. We don't need to do any PCR process. So that's why we require a large amount of the starting material. And for both two kits, we have 2 different options for the live throughput experiment or high throughput experiment. The difference is the indices. For this kit, we provide 24 indices. That means you can pull in 24 samples together in one library. And here we provide the 96 indices, so it's for the high throughput preparation. Okay, so now let's move to the second method to prepare the DNA library. This method, we don't use the mechanical shearing to get the fragment. We use the enzyme, special enzyme called transposon. This enzyme can simultaneously fragment the genomic DNA and add the text at both ends, at its 2 ends. So we call it process called tegmentation. That means fragmentation together with adding tag. And then we use some PCR step to hybridize this adapter. We don't add the adapter via ligation by using the PCR here. And then after several PCR amplification cycle, we get the sequencing ready for fragments. Still have a DNA insert in the middle and the 2 adapters at the 2 ends. So this the Nestera kits, you can finish the whole workflow faster, and it requires a smaller amount of the input DNA samples. For Nestera, we have two options. It depends on your application. The first one called Nestera DNA. So if your target sample is some large or complex genomes, for example, human genomes, mammalian plants, so you can use this one. And if you like to study some bacterial, virus, or some these are amplicons, small plasmids, you can select the Nestera XT. Nestera XT requires small amount of input, only 1 nanogram is enough. And for Nestera DNA, it requires 50 nanogram. And for the sample normalization, Nestera DNA, you need to use bioanalyzer to quantify and some qPCR. But your Nestera XT, it already includes some bead-based normalization method. So you can finish this step very fast and very easily. So both two kits can index as more as 96 samples. Actually, the Nestera XT is the fastest library preparation workflow from Illumina. So when we compare the first two series, one is Nestera, the other one is TrueSeq. So when should I use Nestera? When should I use TrueSeq? For Nestera, if you care about the speed, the duration time of your, the turnaround time of your sequencing experiment, and you care about the ease of use, or your input sample is a bit low, you can try to use Nestera. If you very care about the coverage, uniformity, and the sequencing results, for example, some gaps, you need to reduce the gaps. I suggest you use TrueSeq one because it's the most, it's the widely used method and the classic method to prepare the library of DNA, and the cost may be more economical than this Nestera. So it gives you some suggestion. And for some of the applications, for example, de novo sequencing or some haplotype basing study, the long sequence reads will benefit the final construct of your genomic DNA. So for those kind of applications, we also provide two different methods to construct, firstly, the very long sequence read. The first method, we call it synthetic long read. So here we also get a genomic DNA, and we make some fragmentation, but each fragment is a bit longer. It's not just several hundred base pair, but 10 kilobase. This is what we call the long sequence read. And then we dilute this fragment with 10 kilobase into individual well of 384 multi-well plates within each well, and then to some PCR to amplify these fragments, and then we use the segmentation Nestera transposer enzyme to further fragmentation this 10 kilobase large sequence into some small sequence. This small sequence are ready for our SBS sequencing. But all the reads from each individual well, we already add the adapters with some indices to identify what is each small sequence comes from which individual well. Then finally, we mix all of the small sequence together into our library and do the sequencing. After sequencing, we firstly identify where does each sequence comes from. For example, this sequence, they have the index with for example, the yellow light, so we can see that it comes from this well, and then we firstly construct we first construct all the small sequence from this well and get this 10 kilobase large sequence from this well. And then we, in the second step, we construct the whole genomic from each larger sequence from different wells. So in this way, we finish the construction after 2 steps. So it's we call it synthetic long reads. And the second method is we call it NextSeq mid-pair workflow. Firstly, we use the NextSeq enzyme to break the genomic DNA to get some still very long sequence, for example, several kilobase like this. And then at the 2 ends, we add some adapters here, some tag with the biotin, and then we get this several kilobase long the sequence, and then we circulate it and further fragment, recircle, and we use step testing to specifically capture only this fragment with the biotin label, and then we will add the adapters to this fragment, sequence this small fragment, and then we will know the information of the 2 ends of these very long reads. And then together with the other small sequence, we can firstly construct this large fragment, and then we further construct many large fragments like this to our genomic DNA. So still 2 steps. So so far, do we have any question of the DNA library preparation method? Okay, so maybe let's go to the next part, the RNA library prep. So the RNA library prep. Firstly, RNA sequencing is becoming more and more popular these years because RNA sequencing has many advantages over the conventional method, for example, PCR or microarray. Because RNA sequence can provide you both the qualitative and quantitative method. For example, after the sequencing, we align all the sequence to our reference gene. We will know the sequence of each transcript. But at the same time, we can further use the RNA sequencing to quantify the reads across different samples. For example, this case sample and this control sample. We can obviously see the counting reads of each transcript is quite different. This way, we can quantify the relative expression level of DNA transcripts in these different samples. Is why we call it quantitative. And the second application is RNA sequencing is applicable to all nearly any species. For example, if your research project targets to the human genome, I think you have already got a lot of information. You can, based on this information, to design some specific microarray for your research. If your research is trying to sequence, to get some transcripts from the other species, for these species, they are rarely studied by the researchers, so you cannot find a workable microarray with all of your needed information. But for RNA sequencing, you can just sequence this among transcripts, you can get what you need. So it's not depends on the availability of some microarray products. And then the third advantage of RNA sequencing is it requires no prior knowledge. Suppose you're a supervisor, you ask your students how is some transcriptome like. So you can ask this question in a multi-well question way. So the student will answer, but the answer is what you need. You can also ask this question in an open question. So the students will after he did some experiment, he will answer you something new, something unknown, something interesting. For RNA sequence, it is like an open question. For example, for the alternative transcripts, so we know the RNA processing will remove some of the introns. So remove some of introns, and then the exons will be translated to different proteins. For some of the alternative splicing, for example, this one is already known. So maybe you can design some use some microarray products to sequence this part. But suppose there are some alternative splicing there are known. You cannot find these products. So only based on RNA sequencing you can find this novel translation. So it's something only RNA sequencing can provide. And also some gene fusion. Gene fusion, some specific locus of the gene will be translocated to the other locus within the same chromosome or to the other chromosomes. In this way, they will result in some special gene fusion, and this gene fusion will be translated to some maybe some mutant proteins. These proteins will drive in maybe some cellular dysfunction. But we can only see there is some level of randomness for the happening of this gene fusion. By using RNA sequencing, we can sequence this special locus where the gene fusion may occur. And this way we will know some gene fusion exists or not. And the third example is the legal specific expression. As I said before, RNA sequencing is quantitative. So we can use RNA sequencing to find the unbalanced expression level of some allele genes from the maternal origin or from the paternal origin. So in this way, we can study the genetic variation which is related to the phenotype difference. So that's why RNA sequencing can provide us. So for the RNA library preparation products, we have a very large family of products from the messenger RNA to total RNA, small RNA, and even some target RNA as when you customize your own heptacon. So let's start from the mRNA and the total RNA. So we provide different ways to prepare the libraries. So here we can see the first tool, the TrueSeq mRNA. We use some oligo with poly-T tailing. This way we can capture the messenger RNA with the poly-A tail. Capture this part of mRNA, make some fragments, make cDNA, add adapters, and ready for sequence. This is the first method. We only capture the mRNA. The second method is we also start from total RNA, but here we also use some specific oligos with some specific sequence, and these oligos will target the ribosomal RNA, and then we use some beads to remove this ribosomal RNA. In this way, we remove only the rRNA from total RNA, and we keep the other RNAs from mRNA to some untranslated region or some non-coding RNA, and still make cDNA, add adapters, and ready for sequence. So very simple question, which one do you prefer? Which one do you think is a better way to prepare the messenger RNA sample? Okay, so I would like to say there's no perfect method, of course. So maybe we can first see the disadvantage of the first one. The first one actually is very simple, but it has some limitation. If your RNA is very fragmented, for example, you get your sample from the FFPE samples, the RNA are very fragmented, only a small amount of the messenger RNA has the poly-A tail. So the messenger RNA who are close to the 3' end will be captured, who are far from the 3' end. Because the fragmentation, they won't be captured and won't be sequenced. You'll miss quite a lot of information. So this method is workable for some samples with good quality. Then this one seems better. So we can see some example. For example, the first example, first experiment, we use the messenger RNA method, library preparation. We capture the exons which are close to the 3' end. They have a good sequencing coverage. But this region which are far from the 3' end, we get a very low coverage of the sequence. But if we select the total RNA method, it can give you better results. Both the messenger RNA from close to the 3' end and to the 5'. It's the first advantage of the total RNA method. And the second method, because and here, messenger RNA way, we only find some exons. We only find some coding regions of the RNA. But here, because we use the total RNA, it just removes the ribosomal RNA, but keep the other parts. So not just the coding region, but also some non-coding region. So using the total RNA library preparation, you will give us more information here. Like we can see the upregulation of this expression across the 4 stages. Here, by using mRNA kit, we cannot find this information. So for the total RNA options, we have different kits. So depends on your research. The first one, this kit will remove the cytoplasmic ribosomal RNA from human, mouse, or rat. And this one, it not just remove the cytoplasmic RNA, but also remove this mitochondrial RNA here. And this one, additionally, remove the globin mRNA from the human, mouse, or rat samples. This one is used for the plant. And this one, epidemiology, so it is very compatible with the human microbiome or some bacterial infected human samples. So it will not just remove the cytoplasmic RNA, rRNA and some mitochondrial RNA, and also remove the RNA from the bacteria or some virus. So it's very suitable for the epidemiology study. And these 2 kits, they remove specifically the rRNA from bacteria or some yeast. So we have different options, depends on your research project. For both the 2 kits, mRNA and the total RNA kits, they require maybe different input, different sample input. And this total RNA kit, as I said before, for FFP sample, because it's really highly degraded, so total RNA will help us capture as more as the messenger RNA. And both 2 kits, they support the multiplex of 96 samples. Yeah, both of them use the gel-free protocol, some base size selection protocol. And both of them are compatible with all Illumina sequencing platforms. And then I would like to highlight the other features of these 2 kits. So these 2 kits, we call it stranded kits. They can offer the transcript specific info. Let's see this scheme. So yeah, so if we care about the strand specific information, we only need to sequence the first strand cDNA because it's the coding strand. It's the coding strand. But the second strand of cDNA is the nonsense cDNA. We don't like to sequence this part. We can, by using these 2 kits, we can follow in this way. Well, firstly, we get the fragmented rRNA, and then we do the first reverse transcription, get the first strand cDNA. And in the second step, we incorporate the DUTP instead of the DTP. So the second strand cDNA has the incorporated DOT here. And then we ligate the adapters and followed by some PCR process. In the PCR process, we introduce some high fidelity tag polymerase. This polymerase cannot pass through a strand which incorporates DOT. So in this way, the second strand cDNA will not be amplified. Only the first strand will be amplified. Because the sequence relative to the P5 and the P7 adapter is confirmed, the strand-specific information will be retained in our libraries. So it's our options. So the method based on the DOTP is the widely accepted method to prepare the strand-specific RNA library. So these features can also benefit our research. For example, the first one use some standard RNA-seq library kit. They discover something. There's one intron part between two exons. They can see the reduction of this expression level increase. And then they guess it comes from the expression level of the introns. But if you use some stranded library preparation kit, you can find something different. Here, in this experiment, they find this expression is not just come from the introns expression, but also comes from the expression from the antisense, the second cDNA strand. So that's why we say the stranded library preparation kit can provide much more information compared to the standard one. But our Illumina, we provide both two choices to the users. You can select either the standard one or the strand specific one. Okay, so this slide we have seen in a previous slides. So the mRNA solutions and total RNA solution. So for this one, we know it may face some limitations if your sample is FFP samples. But this one, is still the perfect one. Let's say there's not any perfect library preparation. It's only the most suitable one. For these kits, it has some problem like we only remove the ribosomal RNA, but there are a large number of the non-coding RNA still has been kept in our library. So this part of a non-coding RNA will cost a lot of reads in our sequencing process. That means maybe it requires us to sequence more to cover all of our target coding RNA part with the sufficient depth. So it requires much more sequence, of course, more cost. So we launched the third method for the users. So if you find the limitation of these two methods, you can try to select the third one, but it's a bit more expensive, of course. So we call it RNA axis. So in this method, we firstly start from the total RNA and make cDNA from all of the total RNA and adapters. Then we use a set of specific sequence oligos to capture only the mRNA part. Different to this method, it only has some oligos with poly T tail. This set of probes, they have specific designed sequence. This sequence will capture all of the mRNA part. So in this way, it can effectively capture the mRNA, no matter the RNA sample is fragmented or not. So this kit is extremely helpful for the FFP sample sequencing. And because the capture efficiency is very high, so it requires a small amount of the sample input, only 10 nanograms compared to 100 nanograms for the other two kits. So let's see the comparison of the result. compared with the total RNA kit. Total RNA kit, if you have a 250 mega reads, so for your target mRNA part, you can have a bit higher coverage. But if the total reads is very small, so you cannot get the sufficient sequencing depth. Using the RNA axis, because we specifically target our coding area, so we can get a very good coverage for this target region. So when we compare these two kits, for total RNA, less than 20% is the coding area because they just remove the rRNA. For the RNA axis, it should have more than 70% will be targeted to your coding area, so it's more specific to give you a higher coverage. So this RNA axis is extremely helpful for some gene fusion study. When we compare with standard mRNA kits, we need smaller reads to get enough coverage. And we already launched some pan-cancer panels based on this RNA axis for the RNA sequence. So if you are interested, we can talk more about this kit. Okay, so finally, a very simple slide is for solutions for the small RNA. For small RNA, we add the adapters not to the cDNA, but to the two ends of the small RNA here, and then to the reverse transcription PCR to get the first strand cDNA, and the PCR amplification to add these two adapters. And here we use the gel purification, not the gel-free selection, but the with gel selection to get our microRNA libraries. So the first part is the target library preparation kit. So exon is one of these target library prep options. So to capture the exons, we use some enrichment methods, like the RNA axis. We have some specific probes to target the exon region. And we also have some amplicon method. This way, users can customize your own kit, only target your interested amplicons. So there are two methods. And finally, we also provide a family of fixed panels. We call it TrueSite. They target two different disease-related genes. For example, some cancer-related or autism-related or some inherited disease-related. So let's see the exon kit first. So why we need to sequence the exon? Exon only occupy less than 2% of the human genome, but it contains more than 85% of the disease-related region. So it becomes more and more popular. We can see from the paper citation, we can see the trend. And to sequence a human exon, normally you maybe 5 gigabase data throughput is enough. If you like to sequence a whole genome of a human, it requires at least 90 gigabase. So we can see the exon is a more economical way. And because the data you get is much smaller, the analysis and the stored data is more simple. So that's why people select exons sequence rather than whole genome sequence. We just launched these two kits in the last quarter, TrueSeq exon and TrueSeq rapid exon. So these two kits, they have very good quality, but different features. So you can select either one based on your application requirement. So the first one, it takes a bit longer time. You need to, it takes 1 day to prepare the library, 1.5 days to finish the enrichment. So total 2.5 days to prepare your exon libraries, followed by the sequencing. So the total workflow may take 4 days. 4 days. And for the second options, you can finish the library preparation within 1 day, followed by the sequencing the second day. So you can finish the whole workflow maybe 2 to 2.5 days. So it's a bit faster. To select which one, it depends on your requirement. For example, if you are new to Excel area, you can use the TrueSeq Rapid. The startup cost of this kit is a bit lower, and the turnaround time, the total workflow can be finished within 2 days, and it requires lowest DNA input. But if you prefer a classical way to prepare the library, or you use the FFP samples, you care about the total cost of your exon sequencing. You care about you prefer the highest data quality rather than the turnaround time. You can select the TrueSeq exon. And finally, if you only target on some medically relevant genes, you can select TrueSight 1 panel, fixed panel, or the other inherited disease or some cancer panels. I will introduce in the following slides. Let's compare these two. So they have different requirements on the DNA inputs. For the fast way, the TrueSeq Rapid, it requires 50 nanogram DNA input. This one requires 100 nanogram. It's compatible with FFPE. Both the hands-on time and the total library preparation assay time is longer. But it has a better on-target percent and better coverage of your targets in comparison to this one. This kit is our old kit. You can still purchase this one, but you can compare the features and also the cost with the other two. The TrueSeq exon, here is the US list price, but the price in Hong Kong may be a bit different. I can tell you now, until the end of March, we have a very attractive promotion price for those two kits. So you can contact our account manager for more information. Those two kits, they are compatible with all of our systems, but this one is FFPE compatible. This one is not. Yeah. So both two kits recommend 2x75 base pairs sequencing. We also support 2x100 base pair, but we still recommend 2x75. So when we compare the on-target reads, that means from the final reads lower. For the TrueSeq exon is even better than this one. All of the on-target reads are larger than 80%. Then we see the sequencing coverage of these two kits. Of course, the coverage depends on the total data. Suppose we get the final data 8 gigabase, so we can see nearly 90% of the region will be covered at 20 times. For these kits, it's even better. More than 90% of the region will be covered more than 20 times. So the coverage of comparison between these two kits. And in this data, we use some standard samples from NIST. We would like to compare the variant calling from our exon kit to the database. So the precision, that means a probability at a called variant is correct. That means it's a true variant when we compare our result to the database. It's 99%. Both two kits larger than 99%. And the recall rate, the probability of calling a validating variant, approximately 90%. So it's the performance of these two kits. For FFP samples, as I said, they are more challenging. Using our exome kits, here's a normal sample, so we can have 85% of the on-target rate. But for FFP sample, when we do the test, we can also still get this value very close to 8% on-target rate. So we would like to compare our kit with another kit from the other provider. So both two kits use when we both use 100 nanogram inputs, the on-target rates from our kit is much better than the other one. The coverage depth is also very comparable. So here's the content information of our kits, of these two kits. So according to the standard from the other institutes, our kit can cover more than 99% of the exome region. So the total target size is 45 megabytes. So it's a bit more compact than the other choice. For the TrueSight fixed panel, we have a 12 megabytes. So a more compact library will benefit your throughput. For example, if you use NextSeq instrument in a mid throughput mode and a high throughput mode, if you would like to sequence your exome with the average depth 50 times, then you can get the 4 GB per sample per round, 4 GB per round, and then in NextSeq you can prepare 5 or high mode 15 samples, more than the other one. So finally, we have some validated data from third party. So in this paper, they compare 4 commercially available exome platform, and they said this our 45 megabytes content is very relevant to the human exomes. So as we can see here, the whole exome sequencing is approximately 5 times lower cost than the whole genome sequencing. So it's a good replacement. Here 85% of genetic variation found in our coding regions. So the second method for the target sequencing, the first one is we use for the exome, we use some specific sequence probe to capture the exome region. The second method is we use some amplicon method. This amplicon you can customize by using our tool like the Design Studio. So we will give you a set of pairs of the custom probe. So each probe they are flanked in each region of interest. So here you don't need to fragment your genomic DNA, and this probe will hybridize to the flanking regions, and then followed by the extension and ligation. You can copy your region of interest, and then use PCR to add these adapters, and finally you get your target amplicons here. So it's a product called the TrueSeq Custom Amplicon. So it's a customized choice. This TrueSeq Customized Amplicon, we have actually 2 options. One is we call it version 1.5. The other one is called low input. So you can see from this scheme that the TSA version 1.5 seems more flexible in terms of the amplicon length. It supports 425 base pair and even larger amplicon multiplexity. But this low input, it requires lower input. For example, just 10 nanogram FFP sample is enough. It's compatible with FFP. So 2 different choices. This low input version as low as 10 nanograms. So actually this number is still a bit conservative. For 1 nanogram, you can still get your result. FFP sample, of course, it requires a bit large number. The the total library preparation time is just 6 hours, but hands-on time 3 hours. Improve the workflow, which is automation friendly to some third-party robotic handlers. And also we provide 16 samples, smaller kit size. So if you don't have many samples, you can use our smaller kit size. So it supports more than 1,500 amplicons library, multiplex 96 samples, and fully customizable. So the TSC as a workflow, here you start from the first day. After 6 hours, you can finish the preparation of the libraries, followed by the sequencing on MySeq or HiSeq or NextSeq, and then you can finish the experiments at the end of the second day. So it's a 2-day workflow. So for the besides the target DNA, we also have some target RNA workflow. Very similarly, firstly we reverse transcription RNA to cDNA, then also add these customized amplicons. Use PCR as vectors. So the only difference is we firstly need to convert RNA to cDNA. So similarly, we have customized choice, fixed panels, add-on selection, and different selections. So for targets, we also have some fixed panels. So these panels is very helpful for some translation medicine study. They have fixed gene targets. So for example, the TrueSight 1, this panel is very large. Actually, it targets nearly 5,000 genes associated with the already known clinical phenotypes. It's a very good tools for the screening of the patient sample. So this kit is widely accepted globally. And this kit, TrueSight inherited disease. So it covers all the exons, antral exon boundaries, and the other regions of the 552 genes. So these genes contributing to severe pediatric disease, inherited disease. And this one is to some 100 genes related to the autism or developmental delay. And to cite cancer, 94 genes, and 280 for sleep site, and to cite cardio for some cardiac conditions, disease. So I think for research, maybe this kit will be helpful. So for all the for these TrueSight panels, we have a workflow, 3.5 day workflow. So finish library preparation within 1.5 day, sequencing, and get the reports. But of course, all of our panels, they are only for research use, not for clinical. So as an example of the inherited disease, we collaborate with Dr. Stephen Kingsmore from Children's Mercy Hospital to design this panel. This panel was originally based on 448 disease panels developed from his group. So they are expert design targets. The total library preparation workflow starts from Nestera DNA fragment. Then we get this library. This covers all of the DNA area, and then we denature double strand from each single strand, and then we use a set of biotin labeled probes to capture only our interested regions, and then use traveling beads, magnetic beads, to put on these target regions, and finally get our targets library. So this workflow requires as little as 50 nanogram DNA. So you can finish the library preparation followed by enrichment in 1.5 days, so very fast. Let's see its performance. So the target region size 2.25 megabytes, 552 target genes. So recommend mean coverage of 100 times, and the target minimum coverage 20 times. So if the mean depth of coverage is set at 100 times here, so from this figure you can see more than 97% of the target exons will at least have the coverage greater than 20 times. So here's the coverage uniformity, coverage performance. So the other final part, the other library prep options. So because of the time limit, I don't want to go more details, but the major study is the epigenetics. So here we know that commonly studied epigenetics is the DNA methylation and also the DNA interaction with the proteins. So we have two different choices, methylation and DNA collaboration with the proteins. So Qverview. The Qverview, we fragment the chromatin, and we use some special designed antibodies, some method to pull down these proteins. These proteins could be histones, could be some transcription factors. After we put on this part, we separate these proteins to the DNAs, and then we repair the DNA, add adapters, and sequencing this part of DNA. And for the methylation study, we use the classified to convert all the unmethylated cytosine to uracil, followed by the sequencing. And then the other part, we don't do the bisulfite. Then we can compare the bisulfite convert sample with the other one. Then we will detect the methylation site, just like a single base variant. Finally, I would like to briefly introduce an automated library preparation system. So until now, maybe you will realize that this library preparation is very laborious, very painful, because we said library prep is actually considers multiple steps. You need to use maybe 1 or 2 days to manually prepare your samples. So it requires a large number of different equipment and different operators. So this work can be done by introducing some automatic robotic machine. This machine will free you from some tedious hands-on, and it's more reproducible with the manual operation. But still, you will see this large instrument will be very expensive and complex to learn and operate. So we have another options for your reference. So this is a very small, very cute machine. We call it a NeoPrep. So you can only take 30 minutes. Only need to take 30 minutes hands-on work, and all the other work can be done within this machine. So it can provide the library, the sequencing ready libraries within 10 to 11 hours. So for each round, it will run up to 16 samples, and it requires actually even smaller number of the DNA or RNA input. So until now, this machine only supports 2 of our kits, but in the coming year, I think more and more kits will be launched compatible with this machine. So now these 2 kits, they are TrueSeq nano DNA, the classic one, and the TrueSeq extended mRNA by using the poly A tail to capture the mRNA. So this kit for manual work, we recommend 100 nanogram, but using this machine, only 20, as low as 25 per nanogram input is enough per sample. This one is also 25 nanogram. For manual one, we suggest 100 nanogram. So let's compare the manual work for this stranded mRNA library prep. It takes 1.5 days, hands-on time 4.5 hours using NeoPrep,
30 minutes hands-on and 10 hours total assay time, much faster. And this machine is actually based on very attractive technology called the electrowetting. So when we change the voltage, we can change the surface tension of the surface feature from hydrophobic to hydrophilic. So in this way, we charge each area individually. We can make the droplets move, separate, or merge together with very interesting technology, very reliable. So here is the animation. So all the droplets can be automatically controlled to move, to merge, and to separate. So it's not based on a hydrodynamic way to add some pressure to make it move, like using electrical charging. So it's very reliable, and the machine can be done in a very small footprint. Okay, so let's see its performance. We compare the NeoPrep with some manual processing. But for NeoPrep, we use a smaller amount of the DNA input. They get very comparable coverage. And then we compare the concordance of the library provided by manually and automatically. So this time we only use 25 nanogram, so they showed a very reproducible results. And with using the NeoPrep itself, we would like to compare the reproducibility round to round and the lane to lane. This is round to round, very good concordance. And lane to lane, by using two lanes, the same sample shows a linearity close to 0.998, so very reproducible. So finally, is a final concern maybe is the cost. So the cost, this single table tells you that to select NeoPrep, the capital instrument expense may be a bit higher. In a long time, the reagents and labor and consumable costs will be a bit lower. But if you select the robotic machine, the capital expense is really huge. So if you have more and more samples, of course, both the robotic and the NeoPrep, the average cost will decrease. But still, we can see no matter how many samples you use per year, the NeoPrep cost is much lower, still much lower than the robot. And when we cross this line, maybe it's more cheaper than the manual way. So yeah, thank you for your time. So maybe it's a bit too late. So we are happy to take any questions during a lunch time or in the afternoon. Thank you, folks.
Do we need to depose the sample first? Before the sample?